Alright guys, we are going to pick up where we left off in class. I'm going to do my best to draw for you and try to make this as easy as I can. So we were discussing the G-coupled protein receptors in class, and we were talking about the structure of it. And before we get into the intracellular components, I want to talk a little bit about this binding pocket right here. So we're going to focus on this, and we're also going to focus on, I'm going to try to do the best I can to highlight this. We're going to focus on the inside of this binding pocket here, right? And we're going to talk about how drugs are attracted to this binding pocket and how they actually bind and how they induce their intracellular signaling cascade, which will then create some sort of reaction within the cell here, and then hopefully deliver a therapeutic effect. So we know that the, let me change my colors here. We know that this G-coupled protein receptor is a 7-transmembrane protein. So we can see here that there is 1, 2, 3, 4, 5, 6, and 7. And this protein goes through the membrane. It's a transmembrane protein. It goes through the membrane 7 times, and that gives it one of its most unique characteristics. Now, interestingly, each one of these units here that stem through the membrane is something we called an alpha helice, and this is what we discuss in biochemistry. We're not going to talk about it too much here, but just understand that every time one of these subunits move through the membrane, it's doing so as an alpha helices. Okay, and it's a pretty complex biochemistry that makes these shapes, but it's just something that you need to understand. So this is how that actually looks moving through the membrane. Now, we're going to talk about this binding pocket here, right? And this is the N-terminus of the protein. So I'm just going to say N here, and this is C here. So when we're looking at this binding pocket here, or the N-terminus of this protein, we're going to notice a couple of things. When we're looking at the inside of this binding pocket where a ligand is going to bind, we're going to look at the amino acids that line the inside of this pocket, right? And there are a couple of different amino acids that are really, really pronounced in this binding pocket, okay? And I'm going to show you what those are in a moment. But we know that since this is a protein, it is made of amino acids, and based upon the characteristics of those amino acids, it's going to attract other amino acids with unlike characteristics. So if you think of a positive and a negative, they're attracted to each other. A positive and a positive, they're not going to be attracted to each other. This is not a biochemistry lecture. I just want to show you how drugs are delivered and how they bind to these binding pockets. So on the next section here, I'm going to show you some of the most pronounced and identified, here, and I can even go this way for you, the most pronounced and identified amino acids that are within this binding pocket. So in a G-coupled protein receptor, there is an abundance of amino acids such as aspartic acid, which you can see right here, right? We have some, let me get something that shows up a little more. Aspartic acid, right? We can see that right here. Glutamic acid, these are amino acids, right? You see that right here. There's a lot of serine in there. Serine is very robust inside of the ligand binding pocket. Tyrosine, and we see something called tryptophan. Now, again, this is not a biochemistry lecture, just trying to conceptually show you how a drug will bind to these amino acids that are very rich inside of the ligand binding pocket. So I'm going to turn this way so we can see this receptor and this binding pocket that is essentially awaiting a, get this color here, awaiting a ligand to come in and to bind. Now, why is it important that scientists understand which type of amino acids line the inside of this binding pocket? Well, because they want to deliver a drug. And within this drug, they're going to somehow make sure that the drug can bind to the ligand pocket and stay in that ligand pocket for a period of time so it can deliver its therapeutic effect and its therapeutic message. Now, how do drug designers then get this drug to stick to the ligand pocket? Well, they do a lot of it through first and second pass metabolism. So as we've talked about, first and second pass metabolism of drugs modify the drug, or they cause biotransformation of the drug, or they cause some sort of activation of the drug. Now, we only talked about oxidation and hydroxylation, but there's many types of reaction that will make this drug, one, more soluble so that it can leave the body, it can be excreted, but also so that it can be activated in a way where it can communicate with some of these amino acids that are in this pocket here. So let's look at one modification that might take place, and we've talked about this quite a bit, which is hydroxylation. Well, whenever we have a hydroxyl groups like to form bonds with serine. So if we have a pocket, a ligand pocket, that is robust with serine amino acids, well, then we know that the drug is going to interact and form some type of bond, whether it's a ionic bond, a hydrogen bond, a van der Waals forces bond. Somehow it is going to interact with that pocket and act as a lock and a key. So once the hydroxyl group interacts with the serine group, it is going to be a lock and key relationship, and now that drug is going to be able to either activate or inactivate the ligand pocket associated with one of the G-coupled protein receptors. So likewise, let's look at another modification. Another type of modification that can occur would be adding an aromatic ring to a drug. Now, an aromatic ring is going to communicate with other aromatic rings, and in this case, tryptophan is going to interact with the aromatic ring that has been placed on the drug. So tryptophan also has a very similar aromatic ring, just as the modification on this drug here. So tryptophan, likewise, let me get a pen here. Likewise, has a very similar sort of aromatic ring, and these two rings will be attracted to one another, and they call that T-stacking. So if a drug has, let's say, either it has a hydroxyl group and an aromatic ring, well, now we can assure that the drug is going to bind to two of those robust amino acids that are inside of the ligand pocket. Now let's just go over one more so that we have just more examples to kind of really get you guys to understand this. So one more modification that could take place is an amine group can be attached to a drug. And amine groups like to interact with aspartic acid. So they will form hydrogen bonds. So now, if we add these different groups to the drug, whether they're added prior to ingestion or if modifications occur to a drug, and I can't say this happens to this drug because every drug is so different, but just understand conceptually that these drugs will have amino acids woven into them, into their design, or they will be modified in first pass metabolism, and all of these different modifications will interact with the amino acids that are inside of this binding pocket. So that's how it sticks. Now, interestingly, let me close this off. I'm going to clean all this up. Let me get rid of this. 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 Interestingly, with G-coupled protein receptors, let me get rid of that aromatic ring. Give me 1 second here. Unhide. There we go. And we're going to unhide this. There we go. And we're going to bring that up. So when norepinephrine and epinephrine bind to G-coupled protein receptors, they primarily use hydroxyl groups that are also very robust inside of the binding pocket. So for example, let me see if I can find this here. For example here, some of these most of these G-coupled protein receptors also have robust amounts of hydroxyl groups. Now, these are the mechanisms in which epinephrine and norepinephrine will bind. So I'm just going to show you epinephrine first. Here is the chemical structure of epinephrine, and you will see that epinephrine will form hydrogen bonds with hydroxyl groups that are inside of the ligand pocket. Likewise, you don't have to memorize this, you don't have to draw this, I'm just showing you mechanisms of action. Likewise, norepinephrine does the exact same thing. These hormones, when they're released by the sympathetic nervous system and they are secreted and they bind and they target specific G-coupled protein receptors, well, they're going to interact with the ligand pocket by the hydroxyl groups that are both on the ring of epinephrine, like you can see here, right? We can see, oops, we're going to hide that. Oh my goodness. Shut this off. Let's go back here. Give me 1 second. I apologize. We could see that these groups and these groups will bind together. They'll form hydrogen bonds. These groups and these groups will likewise form hydrogen bonds. And this is how epinephrine and norepinephrine will bind to the ligand pocket and create either an inhibitory or a stimulatory effect that is downstream in the cell. So let me pull up my next set of drawings here and we'll talk about what these downstream effects are. Okay, so now we're back. We're looking at the G-coupled protein receptor once again, and we're going to talk about some of the properties of activation for this receptor. So just for the sake of this conversation, so that we do understand which G-coupled protein we're talking about here, we're going to identify this one as a GS stimulatory G-coupled protein receptor. So that's going to interact with some of the adrenergic receptors that we talked about, and it's going to induce a response. So first and most importantly is that about 50 to 60% of all drugs target G-coupled protein receptors. And we know that we know that the G-coupled protein receptor can either have a, where did this go? Why is that pen not working? Let's try it one more time. We know that this G-coupled protein receptor can bind a drug, right? We know this drug can be an agonist, or it can be an antagonist, right? We know that it can bind epinephrine. We know that it can bind norepi as well, right? So lots of, and there's tons of different hormones that can bind. There are multiple substances that can bind to G-coupled protein receptors. Most of the receptors in your olfactory bulbs and your sense of smell is G-coupled protein receptors. So when you get a whiff of something and that molecule enters your nose, it's going to bind to something. And most robust receptors in your nose is the G-coupled protein receptor. So it's a very, very versatile receptor, and lots of things can bind to it. But we're just looking at it for the through the lens of drugs, epinephrine and norepinephrine. So once we have a ligand bind to the G-coupled protein receptor, then we got to look at what is happening in the C-terminus inside of the cell. So the reason we call this G-coupled protein receptors is because the proteins in the cytosol are G proteins. So we have a trimeric protein cluster that is composed of an alpha subunit, a beta subunit, and a gamma subunit. And these are G proteins. And the beta and the gamma are fixed to the membrane through these lipid anchors right here, they can be released and they can move, but we're going to focus primarily on the alpha subunit. So why are these referred to as G proteins? Well, because they use a type of energy very similar to ATP, but we call it GTP or GDP, which is guanosine diphosphate or guanosine triphosphate. So these are very robust in all of the cells. All of the cells have a very high level of GTP and GDP, and it's because they are the energy for G proteins. So when a G protein is off and we don't have a ligand binding to it, well, what happens is that we have a GDP that is affixed to the alpha subunit, so it is off. So now what is going to turn it on? Well, if you look here, I have a phosphate. And when we add a phosphate to GDP, it is going to turn on the trimeric protein structure or the G proteins. And again, they're G proteins because they use GDP and GTP to regulate their activity, okay? So no ligand, right? We have nothing binding in here. That means that the alpha subunit is off, and what is attached to it to keep it off and to keep it regulated and to keep it from doing whatever it wants to do is a GDP molecule. So what's going to happen? Well, when a ligand does bind, so let's say we have binding in the pocket, and I'm just coloring this in so you can follow along. Well, the protein, the G-coupled protein, follow along, right? I'm just kind of drawing this so you can see where I'm at. It is going to undergo a conformational change, which means it's going to change its shape. And by changing its shape, it's then going to have the G proteins disassociate from each other. So what's going to happen is once a ligand binds, right here in that pocket, we are going to have a conformational change and an enzyme is going to be recruited. And what that enzyme is going to do is it's going to take ATP, it's going to transfer, let me just highlight this so you guys can follow along, this is heavy stuff. It is going to transfer its phosphate, just like this, and the GDP is now going to be converted to GTP. And that is going to turn the alpha subunit on. Okay? So the ATP right here is going to donate its phosphate. We are going to be left with ADP. And right now I know Trent is saying, well, what is doing that, right? Very inquisitive, right? So what is doing that is something called guanylate kinase. And this is an enzyme that is transferring the phosphate from ATP to the alpha subunit, which is currently off. And then that phosphate is going to turn the subunit on. Okay, so let's look at it one more time. We have let me change the color here. Let's go yellow. We have a ligand that is going to bind. And we're talking about a stimulatory, right? I told you this is a G-coupled protein receptor. Ligand is going to bind. The receptor, follow along, here's the receptor. The receptor is going to undergo a conformational change. We are going to have guanylate kinase, which is sensitive to ligand binding in G-coupled proteins. Guanylate kinase is going to take an ATP. It's going to transfer its phosphate to the GDP that is located on the alpha subunit, and that is going to turn the alpha subunit on. So let's just look at this for a second. I'm going to just cross this out. We are no longer off. We are on, and the conformational change is now going to occur. So give me 1 second. Let me clean up these slides, and then we will move forward with the remainder of this part of the lecture. Let me clean all this off here. Okay. So we had said that the ligand had bound. It had activated the G-coupled protein receptor. We had said that the G proteins are that trimeric protein cluster that is in the cytosol of the cell, and it is currently off. But the ligand binds. Once the ligand binds, it recruits, what is that kinase? Guanylate kinase. That is going to transfer a phosphate from ATP to the GDP that is on the alpha subunit, and then that alpha subunit is turned on. Now, when that alpha subunit turns on, it is going to disassociate from the trimeric cluster. So you can see here that beta and gamma are still anchored into the membrane via those lipid anchors. But the alpha subunit, which now has GTP, it is energized, it is going to disassociate, you've got to remember these words, and it is going to translocate to another enzyme or protein, same thing, in the cell. And that protein that it is going to communicate with is called adenylate cyclase. This is just an intracellular mediator. This is transmembrane. And what this communication is going to do, we will see the active alpha subunit with the GTP. It is going to bind to adenylate cyclase. And once this occurs, adenylate cyclase is going to react. So what adenylate cyclase does is it will take ATP in the cell and it will convert ATP to something called CCAMP, which is also called cyclic AMP. Okay? So CCAMP is a secondary messenger. And what that means is it is a regulatory step where ATP will bind to adenylate cyclase. So you can see it here. ATP will bind to adenylate cyclase. Adenylate cyclase is an enzyme that will convert ATP to cyclic AMP or CCAMP. And I'll just highlight that here. Let's get a different color. Let's go with pink. And we'll highlight there. Now, cyclic AMP is a secondary messenger. So I'm just going to write second. And what that means is it's not the primary signal, it's a secondary signal. So I'm just going to say second. And cyclic AMP is released in very high concentrations. So this is an amplification signal, right? So watch, it amplifies its signal. Now, there's something else that you're very familiar with that does a similar action in skeletal muscle. And I want you to think about what is the largest amplification of the skeletal muscle contraction, or the signal to contract that comes from the alpha motor neuron. What do you think is the amplifier in that pathway? And you should be saying calcium, because we know calcium is released in a very high concentration. So in skeletal muscle, when the alpha motor neuron depolarizes and it tells the skeletal muscle to depolarize, and that signal goes down to the transverse tubules and into the sarcoplasmic reticulum, well, calcium is released. And calcium is an amplification. It is a secondary messenger. By no means is it the first messenger. Acetylcholine is the first messenger. So cyclic AMP does something very similar. Now, where I'm going to stop this is the downstream target of cyclic AMP, because there's lots of downstream targets. And the only one I want to talk about today is an enzyme or a protein called PKA, protein kinase A, and we are going to stop there. So all I want you to know is that for a stimulatory G-coupled protein receptor, what's going to happen is we're going to have a ligand bind to the pocket. It is going to activate the G-coupled protein receptor, which is then going to convert GDP on the alpha subunit to GTP, which is going to turn on the alpha subunit and cause a conformational change. That conformational change is going to be that the alpha subunit is going to dissociate from the trimeric protein cluster. It is going to translocate, and it is going to bind to adenylate cyclase. This is all regulatory steps. Once it binds to adenylate cyclase, the enzyme is turned on, and it is going to start converting ATP to cyclic AMP, and cyclic AMP is a secondary messenger that is going to amplify adenylate cyclase's signal. And one of the downstream targets that cyclic AMP is going to communicate with is pKa. And we are going to stop there because it's going to get even heavier after we talk about what pKa is capable of doing within certain cells, within certain tissues. Okay, so again, a G-coupled protein receptor is an adrenergic receptor. It is going to receive a signal from epinephrine or norepinephrine, or it's going to receive a signal from a drug. That drug is going to be an antagonist, or it's going to be a agonist that is going to modify pKa action in some capacity. So that's all I have for you today. I will see you guys on Monday. Please prepare for your presentations, and we will pick up on this as soon as we're done with presenting. And cyclic AMP is released in very high concentrations. So this is an amplification signal, right? So watch, it amplifies its signal. Now, there's something else that you're very familiar with that does a similar action in skeletal muscle, and I want you to think about what is the largest amplification of the skeletal muscle contraction, or the signal to contract that comes from the alpha motor neuron. What do you think is the amplifier in that pathway? And you should be saying calcium, because we know calcium is released in a very high concentration. So in skeletal muscle, when the alpha motor neuron depolarizes and it tells the skeletal muscle to depolarize, and that signal goes down to the transverse tubules and into the sarcoplasmic reticulum, well, calcium is released. And calcium is an amplification. It is a secondary messenger. By no means is it the first messenger. Acetylcholine is the first messenger. So cyclic AMP does something very similar. Now, where I'm going to stop this is the downstream target of cyclic AMP, because there's lots of downstream targets. And the only one I want to talk about today is an enzyme or a protein called pKa, protein kinase A, and we are going to stop there. So all I want you to know is that for a stimulatory G-coupled protein receptor, what's going to happen is we're going to have a ligand bind into the pocket. It is going to activate the G-coupled protein receptor, which is then going to convert GDP on the alpha subunit to GTP, which is going to turn on the alpha subunit and cause a conformational change. That conformational change is going to be that the alpha subunit is going to dissociate from the trimeric protein cluster. It is going to translocate, and it is going to bind to adenylate cyclase. This is all regulatory steps. Once it binds to adenylate cyclase, the enzyme is turned on, and it is going to start converting ATP to cyclic AMP, and cyclic AMP is a secondary messenger that is going to amplify adenylate cyclase's signal. And one of the downstream targets that cyclic AMP is going to communicate with is pKa. And we are going to stop there because it's going to get even heavier after we talk about what pKa is capable of doing within certain cells, within certain tissues. Okay, so again, a G-coupled protein receptor is an adrenergic receptor. It is going to receive a signal from epinephrine or norepinephrine, or it's going to receive a signal from a drug. That drug is going to be an antagonist, or it's going to be a agonist that is going to modify pKa action in some capacity. So that's all I have for you today. I will see you guys on Monday. Please prepare for your presentations, and we will pick up on this as soon as we're done with presenting.